Hey there, Astronomy Buzz from Lady Brabourne College here. We are going to take you through a virtual rocket ride through the cosmos. We all have heard about Neil Armstrong's giant leap for mankind, but what about microbes? Do they just not exist out there? Or are astronauts secretly sterilizing the solar system with ethanol and cotton? That's what we're here to find out. But first things first, captain before crew. Our work owes everything to the master of us all, the ISS. This orbiting home for astronauts 250 miles above Earth circles the planet every 90 minutes and tests systems for future Moon and Mars missions. Moving on to the experiment that we chose, Expedition 16. It studied how long E. cloacae and A. baumannii, infection-causing antibiotic-resistant bacteria, survived in space and whether such exposure altered their genes that affected their virulence or drug resistance.
While this was our main focus, the mission also examined microbial survivability, growth rates, microbial kinetics, DNA mutation rates, and pathogenicity in space. Now you may ask, why do we even need this in space? Very true. Well, it contributes to the emerging field of medical astromicrobiology, a discipline dedicated to understanding and mitigating the risk of microbial infections in space. As we prepare for longer and more ambitious missions, knowing how microbes behave in space is essential to safeguarding astronaut health and ensuring mission success. In the unique condition of space, microbes may evolve, becoming more resilient and potentially more dangerous. Finally, space condition could accelerate the development of antibiotic resistance, making some bacteria harder to treat upon return to Earth. These factors make our experiment relevant and also critical for the future of human spaceflight. Now, let's peek into what we found in this cosmic microbial gossip column.
In space, biofilms don't just look gross, they can harm astronaut health and damage spacecraft parts. Streptomyces could be lifesavers by making antibiotics in orbit. Some bacteria, like the glowing ones in bobtail squid, behave differently in microgravity. On shuttle mission STS-115, Salmonella typhimurium became more virulent in mice. On the ISS, E. coli tolerated higher doses of gentamicin than on Earth. The Microbial Tracking 2 study spotted Staphylococcus and Malassezia as common space hitchhikers. Roscosmos's Biorisk MSV found microbes that survived extreme space and still reproduced. Anabina, a microbe that scrubs carbon dioxide, makes oxygen and fertilizes nitrogen, is basically a whole life support crew in one cell. Even Sphingomonas and Penicillium could help clean up waste in space.
Okay, so what do we do with all these microbes in space stuff? Well, imagine future Mars bases using algae and friendly microbes to recycle air and water, like ESA's Melissa project. But we have to make sure space bacteria do not crash the system. NASA even tested Sporosaurus unipass theory for space breaks and yeast for protein-rich food. Though, finger crossed, microbes do not eat the food before we do. Tiny passengers. Huge responsibilities. Understanding how spaceflight affect this delicate balance in the survivability of microbes is crucial, not only for astronauts' health but also for us on Earth. How? Well, space changes gut bacteria, which can affect energy use and health.
Some microbes make special chemicals only in trace amounts on Earth. But in space, they produce more, opening doors for new drugs and bioprocessing breakthroughs. Before we wrap up, here's what we'd really love to throw at the scientists just to keep them on their toes. What would happen if microbiota from space shuttles do end up contaminating other planets and celestial bodies? Would it be possible for microbes to evolve and eventually survive under the stressful space conditions if genetically engineered with some armor and gear? If certain microbes do persevere and succeed in natural astrosurvival, what is the possibility that their adapted machinery helps other living creatures inhabit outer space? Before signing off, we'd like to add that if these microscopic voyagers can conquer the stars, then we're just not sending rockets into space. We're planting the seeds of life in the universe. And maybe, just maybe, the next chapter of life's story won't begin on Earth, but out there among the stars.